Okay.
Okay, I see our countdown.
Yep, it's going. Hi, everybody. Welcome to the first episode of our new show. The show is called MVPs, or Making Viruses Pop. And I am one of your co-hosts, Dr. Angela Rasmussen. I'm a virologist, an American virologist who lives in the Canadian prairies. There's actually a pretty nice lab here. And I'm here today with my friend and co-host, Dr. Juliet Morrison, who I'm going to let introduce herself in a second. But Dr. Morrison, Juliet, and I have been friends since graduate school. And we thought that, our field is having a really tough time right now. It's really under attack, and it seems like we're talking about everything except for science these days. And, for going on more than 20 years now, we've been having a really nice time together talking about virology. And so we thought that actually it would be nice to inject some virology into these conversations, because so much of the reason that our field is under attack is that people just don't know that much about virology. So we wanted to have this show to talk about what we really think is very interesting and important about viruses, but in a way that I think is hopefully engaging and fun to follow along with. Because me and Juliet, we had a reputation back in grad school for being among the most fun and desirable to hang out with people. As you can imagine, we're incredibly charming and amusing, and we like to have a good time. So you could always count on a great spirited conversation with us about viruses. Without further ado, I'm going to let Juliet go ahead and talk about her love for viruses and why she wanted to do a show with me after all these years.
Well, Dr. Angie, that is hilarious. I don't think we had a reputation of being fun in grad school. I mean, did we? No, I don't feel that way. But me,
I think
well,
we did.
yeah, we were more social. We went to Free Fridays. We did these things. Free Fridays.
I
by the way,
literally
was
got
like,
elected as like the graduate student president because
you
of
were
my
the graduate student.
funness.
Yes.
And my ability to organize Free Friday, which for everybody watching, graduate students don't get paid a lot. And so Free Friday was Columbia University paying for us to have free beer and pizza. And my reputation is, the life of the party and a connoisseur of beer and pizza got me elected to the apex of graduate student leadership positions.
Yep. And I think you did a great job, actually. I felt like we had always had beer and we always had pizza.
I had a
And
co-president, also a woman, and so you can
Mhm.
imagine that it was
Very
a tight
well run.
ship. The trains ran on time, and we always placed the order to La Familia or
Yep.
whatever. We ordered
Yeah,
the
no, for
pizza
real.
from.
Like, no, it was well done. But yeah, okay. So my name is Juliet Morrison. I am originally from the island of Jamaica, which is in the Caribbean, and I love viruses. I developed that love very early on in life. Like initially I was pre-med when I was like in college, and then at some point like I realized just how exciting it was to do research once I was exposed to it. And I think that's one of the things that a lot of people don't realize is that there's this whole other career path that you could take if you love science. Like you don't just have to you can get into all sorts of things. And so when I went for my PhD, and Angie and I actually started in the same lab, Vincent Racconello's lab, at the same time, same year, and our benches were beside each other's, like across from each other's, actually.
Across from each other.
Yeah, and so we spent a lot of time together, and not only did we hang out in Vincent's lab. years later, we ended up being in the same lab that Angie had gone for her postdoc. So I had gone to a different lab and then got recruited to her lab as well at University of Washington. And so we were able to, like learn more about, like, that was a shift actually from like studying more molecular and I don't know, wet lab work to more systems based computational work. And since then, we have continued to collaborate and talk about science. Like, one of the things that I love about Angie is that I actually respect her as a scientist, but I also respect her as somebody to like hang out with. And I know that we'll have a good time and have great conversations. All right, let's go.
Exactly. Like I said, our reputation for fun. I mean, this has been going on now for, like I said, more than 20 years. And you know what, Juliet? I mean, Friday, talk about feeling, old, because I've been trying to hype this show up as though we are, who we were in graduate school, which again was we were people who had a reputation for being not only fun, but I had a reputation for making some of like the best rap mixes on Columbia's medical center campus, which wasn't really a tall order, but we had a reputation for being the life of the party. Friday was the 17th anniversary of my PhD thesis defense. And you want to know something else? Here's something fun, Juliet. You recall, and for viewers watching, anybody who's been following me on either Twitter or Substack has perhaps heard me make a reference to the fact that my graduate project was not a smashing scientific success. And, this was something that, I couldn't wait to get out of grad school. I mean, I hated my project to the point that, it didn't work. And I finally figured out after 2 years why it didn't work. And then I had to start from scratch. And then it took forever to do one thing after another. And it was just so frustrating. And, 6 years, took me 6 years to get my PhD. It was terrible. And I was just so discouraged by it. But since then, like, I'm so glad that I had that experience, actually, because it really taught me a lot about how to persist through failure, scientific failure. It also taught me a lot about how to be a good experimental scientist. But I think the thing that has been most gratifying was that I was giving a talk at Yale at the end of March. And I met with one of our colleagues there, Ellen Foxman, who's an immunologist in the department that our colleague David Martinez, who's a coronavirologist, invited me. And I met with a large number, or not a large number, but, I met with faculty while I was there. And I met with this woman, Ellen Foxman. She told me that she has read my paper from graduate school, almost more than any other paper, for a project that she was working on.
Wow.
And she thought it was so good. And nobody has done that type of work before. I was trying to make a mouse model of rhinovirus. And we actually got into a big long discussion. And this reminds me, I promised her to send her a PDF copy of my thesis and I forgot. So I better do that as soon as we're done here. But it was just so gratifying to out of nowhere, I picked up the stuff where I left off. And I have been talking to my students about my PhD project as a way to encourage them to, power through tough times.
Yes, because you have to.
You have to, and I sure did for my PhD project. So I'm glad that I did. But boy, can I not believe that it's been 17 years, since then. And it just goes to show that you never know what kind of viruses are going to cross your path and when they're going to cross your path. It was just really delightful to talk about the, molecular basis for mouse adaptation of rhinoviruses or a lack thereof. And challenges in achieving such. which was the main problem with my PhD project. But in any case, I'm out of the rhinovirus game, but it was nice to talk about it. You never know when rhinoviruses are going to be back and relevant. And
we don't ever know. Like, we didn't know that about coronaviruses, that something would like pop up that would actually be sort of a pandemic type thing. So you just don't know. Like, we need to, that's why people study multiple things, because you never really know what's going to actually pop out to be the next problem. Personally, if rhinoviruses became the new thing, I would be so shocked, but also not shocked.
I would be too. Nobody would be more shocked than me. And
their thing.
And boy, would I be deeply annoyed that I have to start working on rhinoviruses again. But you know, that seems to be the story of my life, and that's actually kind of the topic of today's episode, is how annoyed we are with viruses that we shouldn't have to know that much about, because they should be not a problem for us as something to continue researching since we've defeated them with vaccines. And in particular, I mean, so Juliet and I should be used to this, because in Vincent's lab, it was Vincent Racaniello, our PhD mentor, was the first person to clone an animal virus, a virus that was a relevant human pathogen. And that was poliovirus in the late 70s, early 80s, when he was a postdoctoral fellow in David Baltimore's lab at the Massachusetts Institute of Technology, or MIT. This was a big achievement in molecular virology. And as Juliet had mentioned, we both were trained in this very lab heavy environment with a lot of this really classical molecular virology work. And so we've worked, using older techniques. We know about all the history of these, of virology, these old viruses. I mean, Vincent had this cabinet full of stuff, and he still has some of that stuff at his studio, the incubator, because we are chips off the old block, and we are trying to impress our PhD mentor by having our own show, because he is a veritable scientific media mogul at this point. I just want to give a quick shout out to This Week in Virology, his flagship show, but microbe.tv, his entire production media empire, it's
awesome. Has some really good stuff.
Yeah. It's got fantastic stuff about immunology, evolution, parasites, infectious diseases, clinical stuff, of course, viruses. Vincent is Earth's virology professor. And speaking as somebody who trained with him, I'm sure Juliet would agree, he's an excellent teacher. And excellent.
We have always said that. Like,
yeah,
Vincent is really good at teaching. Like, I think we had exceptional training.
We had exceptional training and exceptional teaching. And a whole other topic is we can talk about all the professors that dialed into the classes where Vincent showed up and actually gave us a lecture that I could understand. Yep. And that wasn't a stack of crystal structures of transfer RNA. I still will never forgive that particular guy who taught biochemistry our first.
Oh my God, that biochemistry class was the worst.
You remember it would be like pop quiz, predict the RNA structure.
Yeah, and you're like, but you didn't even teach us anything. You just gave us a stack of random crap. Like, I don't even know what this means.
I complained about it at one point, like, raised my hand and like gave a sassy little like quip. And, the professor was like, well, what do you think you're going to do if you ever need to know an RNA structure? And I'm like, call an RNA structure person. What do you think I'm gonna do? Like,
yeah,
I'm a freaking microbiology person. Like, no.
Like, how would you expect us to do this out of our heads? But quite frankly, like, even RNA people are not doing that. So it was kind of ridiculous.
Well, in this class, so Vincent taught some lectures on translation, on the process of taking messenger RNA, which encodes, it's the genetic code. It basically is how your DNA turns into a protein that goes out and does things biologically. And so the process of making DNA to messenger RNA is called transcription. And the process of taking that messenger RNA which goes out of the nucleus into the ribosome and translates it into a protein, is called translation. And so Vincent taught us about translation because in the old days, people thought they were studying viruses, and then they ended up finding out really fundamental things about the way that cells work. And one of those things was that poliovirus, which Vincent works on, has a really unique and creative mechanism for shutting down the host's ability to make proteins to do translation, but it can do its own translation. And in the course of understanding how the virus did that, we actually learned a lot about translation in general. So Vincent taught a class on it, and it was really great because he actually didn't just like recycle some old slides, which, oh God, I have to confess, I do that a lot. And at least I'm upfront about it though. And I do try to involve the students so that it's more of an interactive learning experience than just getting bitched at for not knowing how to solve RNA structures out of nowhere. Anyhow, I'm annoyed, even though I'm familiar with all this history, even though Vincent worked on a virus that I should not have to know about. Is why I worked on rhinovirus in graduate school, because we all thought polio was going to be eradicated. And I wanted to study pathogenesis because, as you know, I've always been wild about pathogenesis, or the process by which viruses cause disease. This involves a lot of interaction with their hosts. And I study this process more from the host side of things, actually. But you can't really separate the two. It's kind of
you cannot
egg thing. But anyways, this is something I never thought that I would have to learn that much about polio pathogenesis, besides what's already been known, because the polio vaccine works, and we thought it was going to be eradicated. So I thought, oh, I'm going to cure the common cold instead. Yeah, I totally did that. I didn't think that I would have to know stuff about measles virus, for example. And because, measles virus, it's a paramyxovirus. Polio is a different family of viruses. It's a picornavirus. Measles is a negative sense RNA virus. Polio is a positive sense RNA virus. Measles is enveloped. Polio is not. And these things are all like they're big differences in terms of the molecular virology. Like the virus has to do this, these viruses have to do the same things. Generally, they have to get into a host cell, they have to replicate, they have to make new viruses, they have to make the viral proteins, they have to replicate the genome, they have to get the new baby viruses, which we call progeny, out of the cell. All these things they have to interact with the host for, and that happens differently. It happens really differently from one virus to another, even though they're doing the same thing ultimately. A lot of roads to the same destination. And so it takes a long time to actually learn this about all these dumb viruses.
It's a long time. And you still don't even after you've spent 6 years doing your PhD and then doing all this additional training, you tend to narrow things down, so you're not initially you learn about a whole bunch of stuff, and now you've gotten into your own areas of interest. Sometimes they might be different types of viruses. For me, I study influenza, which is a negative sense virus, and I also study dengue, which is a positive sense, but ultimately you don't know. You eventually pick the virus that you like, right? Or the viruses that you like. And you never expect that viruses that are preventable from vaccines, or things that were eliminated in 2000, the year 2000, like measles, that you would need to know that much about them, because they are actually officially kind of gone. And yet here they are, they're back. And so now we have to relearn all of this, or we have to change our perspective and focus on things that we should not have to focus on. And
exactly. Now I need to know about measles pathogenesis, for example. That was, our colleague Paul Dupree has done some beautiful work on measles pathogenesis. And I'm just happy to leave him to that. Like, I don't want to start doing this paramyxovirus stuff. It's not even the negative stranded thing because, I graduated from ambicurious to fully ambisense like years ago. I study Ebola, which is also, and flu, which are both negative sense. But so I got over that. But I think that we shouldn't have to know that much about them anymore besides what makes the vaccines work. Like, these are diseases that would be scientifically fascinating, but here's a great example of virology research that you shouldn't do, virology research that you don't need to do. Because if measles isn't causing epidemics, if we could eradicate measles, which theoretically is actually possible, if we did that, we wouldn't have to worry about measles at all anymore. And any sort of scientific value that we could get from studying measles, we could probably get from studying a different virus. And so measles should be gone and should be extinct. Same thing is true for polio. I mean, eventually we should be able to eradicate polio. But now, I mean, we're going completely backwards. And so that's what I wanted to talk about today was these paramyxoviruses. It's irritating because I'm interested in some of them. Theoretically, I'm interested in RSV as a public health issue, but I'm not really that interested in RSV as a virus. I certainly RSV is a virus that is spreading. So respiratory syncytial virus, causes these seasonal epidemics of respiratory disease. And for most adults and immunocompetent people and even older kids, they've got residual immunity from their first exposure, and they're not going to have a rough time with it. They're not going to get it for many years. And when they get it again, they're going to get an immunity boost. And, they're not going to have a problem with RSV until they get older, and then they have medical risk factors. And so older people and very young people can get access to the RSV vaccines. And there's two, one is antibodies, one is a vaccine. They both work essentially the same way, except the vaccine produces immune memory, whereas the antibodies just provide some temporary immune protection. But that is really important for some people who are very vulnerable when RSV is spreading. And right now, RSV is spreading so much that for people who are eligible for RSV, most states in the US have extended the window of time that eligible people can actually get the RSV vaccine. And the reason I bring this up is because a very close family friend, one of my best friends' family members, got RSV despite having the antibodies or the vaccination. I apologize. She's an older person. She has pre-existing medical risk conditions. And people should also be aware that the vaccine isn't a foolproof protection against getting RSV. What's good is that while she is in the hospital for RSV, she's actually in such good spirits that she is vigorously complaining verbally, which is a great sign for a disease that causes pneumonia and shortness of breath. So it's really important to emphasize that while vaccinated people who are at risk for RSV sometimes can still get it, especially if they have other conditions that would make it more serious or if they're immune suppressed, it still provides a benefit in terms of mediating the severity of the illness.
No, absolutely. Because if these, the way I think about it and how I explain it to my students is that, yes, you got this vaccine, you've mounted antibodies, you have like a cellular immune response, and you would think that that would protect you completely. It protects you somewhat, but actually, if you didn't have this, you'd be so much worse off. So it's better to get your vaccine. And even if you get sick and you're like, I got the flu or I got whatever, even though I had the vaccine, well, probably you would have had it worse without the vaccine. Sounds to me like you're a person who might not have the best immune system. Good for you that you got your vaccine then.
It's really important if you're able to get vaccinated. And this is something that drives me crazy. I think we should do a whole separate episode on this with flu. That vaccines don't work unless they are completely sterilizing. And that's just absolutely not true. Vaccines work by preventing or reducing disease. They don't prevent infection. They were never designed to do that. Edward Jenner, the guy who gets the credit anyways for discovering that vaccinia virus infection could protect against variola or smallpox infection, didn't know what a virus was, so he didn't know anything about preventing an infection. He knew about preventing smallpox, the disease. And that's how vaccines are designed, and that's how they work. So it's really important for people to understand that vaccines prevent disease. RSV will reduce disease. It is very difficult to get completely protective immunity for respiratory diseases. But you know what? There's one exception, and that is measles virus. Because measles is so well adapted to humans that it actually can't really evolve around the vaccines anymore.
Yeah.
And so if you get vaccinated against measles, it protects completely against measles, because measles is not exclusively a respiratory disease, even though it is. So you will not, the vaccine, the MMR vaccine, is 97% effective. If you haven't gotten it, if your children haven't gotten it and they're older than 12 months of age, please get it, because there's a lot of measles out there too. But these viruses are pretty interesting. And I mean, what I really like about this show is that Juliet does a lot more teaching than I do. She is truly a scholar and an educator, as well as, a virologist. I mean, I'm a researcher, and I do have students that I've trained, and I teach lectures sometimes, but I don't do as much in person, lecture style teaching. And I've had to essentially listen to a lecture from somebody like Juliet on YouTube, and look it up in Fields Virology, which is our standard textbook for the fields, to kind of figure out, and Wikipedia, because I actually do that too. But
Wikipedia is awesome.
Wikipedia is great for a broad overview. Yeah, so I actually start with Wikipedia.
Why people put Wikipedia down. I think it's an exceptional way to interact with knowledge, because you can follow up on any of the citations that they provide you. I think it's great.
Well, there you go. I mean, I agree, but there you go. Endorsed by Professor Morrison, a far more accomplished teacher than myself.
Okay. Yeah. Yeah, no, good stuff.
But all about these Paramyxoviruses a lot more than me. I mean, I can bullshit about Nipah, but I've actually not done enough meaningful work myself on Nipah that I've had to go to there's talk to the media level knowledge, and then there's master professor level virologist knowledge. And I feel like I've gone from, as the co-editor in chief of Vaccine, here's my commentary on vaccine level of knowledge, which needs to be advanced for vaccines, but not for those viruses. And now I've got to go to I got to be at Juliet at least, where I can, talk about how Paramyxoviruses assemble replication complexes. And I just don't, I've never been good with the details of all those things anyway.
I mean, I'll be honest, okay, so fine. I teach a bit, I teach a little bit about RSV in my course, but I tend to stick I teach 2 courses at the University of California, Riverside. So I teach comparative virology, where we look at all the viral families and we compare the different replication strategies. And then I also teach a course that I developed myself. It's called Host Responses to Viral Pathogens. So it's basically studying how your immune system at multiple levels, yay,
my favorite topic in the world.
Yeah, exactly. Can either inhibit viral replication, or actually can lead to the progression of viral disease. So a Goldilocks kind of effect. Too much of your immune system is bad, but too little of it is bad too. But one thing I do talk about with my students during the course is RSV for a little bit, as in, where does the name respiratory syncytial virus come from? What are syncytia? And so one of the reason that we have this is because The reason that we've called RSV is because it causes this interesting phenomenon where once it infects one cell, it um, expresses proteins that allow that cell to then bind to other cells, so that you end up with this like multicellular complex that we call a syncytia. And that's where that comes from. And you can very easily tell that RSV is a causative agent of a disease if you see syncytia versus other sorts of cytopathic effect. And by cytopathic effect, I mean the way a cell dies. But the way the cells die with respiratory syncytial virus is pretty cool. Like they form these little blobs of mega cells.
Yeah, but it's annoying because CPE, or cytopathic effect, is way easier to see. This is straight up, like, I hate these syncytia. These syncytia forming viruses are so dumb. I really prefer CPE. Hepatitis C virus, which I did my postdoc on, sort of forms syncytia. Like, they're not very good. And which you'd think that they would be great, considering it's got like 5 freaking receptors and they're all like tight junction proteins and stuff. But no, like, it doesn't really form very good syncytia. It forms like tight junction connections, and it's all the hepatocytes. So you can't really tell until the infection is so widespread, at least in the liver, that like all the hepatocytes are like, malformed and look weird. And they de-differentiate and lose their structure. But like, whatever. I just hate these dumb viruses. It took forever to grow. It was such a pain. I couldn't do a plaque assay with it. I had to do a focus forming assay. A plaque assay is when you look for cytopathic effect and you actually dilute your virus so that you make these little plaques. They're like little holes in a carpet of cells on the bottom of a plate. And you can count those like afterwards. You stain them so that they're purple. And then there'll be little like clear spots, and you count those, and you can use that to figure out how many infectious virus particles you have. We call it a plaque assay because those little spots are plaques. Plaque assay doesn't work for viruses that are not cytopathic. So you can count syncytia under a microscope, or you can do what's called a focus forming assay, where you either have them labeled with some kind of fluorescent thing, and then you take a picture of it and you can count that, or you can do it the old-fashioned way, like I used to do it as a postdoc, where you can stain it with an antibody and then use like some kind of color-producing agent to develop it. There's different ways to detect it, but yeah, I mean, it's good times. I hate those syncytia formers. It takes forever. Everything is like an ordeal. Plaque assay, nice, clear thing. Yeah. Even easier, medium tissue culture infectious dose assay, TCID 50. Mm-hmm. All you got to do is CPE, yay or nay. You got a nice cytopathic virus, boy, who's been a virologist for a while, because, a glance, that's CPE.
Right.
Some of my trainees over the years, they're like, oh, I can't tell. And I'm like, that's why you always have a negative control, because if you stick something with SARS-2, which is a super cytopathic virus, up next to a mock infected like the difference is immediately obvious.
It's so obvious. Yeah.
Like,
it's so obvious.
It doesn't even have to be polio, the most beautiful virus to work with.
The best of all.
The best of the all time. Polio. Oh God, the goat. Like, I'm not happy that it's back, but I guess like it'll come in handy that we have all this polio knowledge. Wow. And even I can't screw up the cloning with polio. It's a very forgiving virus from a molecular biology perspective.
It's so robust. It's the best virus I've ever worked with. And that's
you have to try
why I worked on it in my PhD, because like I know people were like, why are you working on this thing that's almost eradicated? I mean, I was like, because it's robust. Like, what a lovely tool for exploring cell biology. It's very cool. Yeah,
it's a beautiful virus to work with. It's a terrible
virus. It sucks that it's back.
It's a terrible virus. It's a human pathogen. I mean, this is the thing about being a virologist. Like, and I think this has been really misunderstood about our field, is that, you know, I think viruses are beautiful. Vincent got a lot of flack from like the lab leak community because we were both in this documentary about the origins of the SARS-2 pandemic. And Vincent said that he thinks he might love viruses more than his own family. And, but you can hear him saying that, right, Juliet?
Yeah.
Next episode, I'm going to cut a clip of it.
Yeah.
But I mean, both of us were in this documentary, but not only did it not surprise me at all that Vincent said that, but I actually know exactly what he means. And he did not mean that he loves viruses actually more than his family, to be clear to everybody.
Right.
He loves viruses. It's his life's work, and he's done incredible work. He's made incredible contributions, and both of us were so lucky to have trained with him. And honestly, Juliet, like, I don't know how you feel, but I feel like in many ways, like knowing about the old way of doing things from training with Vincent and having somebody as accomplished as Vincent to always feel like I could never remotely live up to like the standard that he set has really prepared me for this moment in particular. To stand up for science. And, you know, as you know, like I have a show with Vincent that, you know, we're doing together, and I think the communication aspect of it's really important. There's nothing more important to me anyways than preserving virology. And I think that a big part of it is helping people to understand like what Vincent means when he says that, or to understand
yes,
what
absolutely.
when they say that viruses are beautiful.
Yeah.
It doesn't mean that I want polio to come back. It means that I
Exactly.
want polio to be gone forever, and like think fondly back on polio like it's my dead boyfriend who died before we could get married.
Exactly. No, but we can admit that there is a beauty to these processes, and a beauty to the symmetry, and a beauty to all of these things. And that's what gets us excited, because like we're learning things about viruses, their processes, but also like the cell itself, which is us, at, you know, at a fundamental level. So yeah, like it does worry me that people, when we say, oh, we love viruses, that people are like, oh, is something off with you? Are you going to be like that kind of, you know, comic book or TV sort of like villain doing stuff like that? No, like we appreciate cool things, and it's pretty cool. Viruses do very exciting, interesting things that can teach us stuff, but also can tell us, I mean, teach us stuff about how we work, but can also tell us about how we can like suppress them when they're infecting us. And I find that, I think it's really fascinating. Like it's what makes the job very exciting. Like you don't go into this because you're like, I'm going to make a ton of money or something. Like you go into it because you're like very excited about learning more about what is going on. And I feel like I feel good over the years, like knowing that like I've contributed to some level of knowledge that's out there. And I bet you feel the same way, Angie. Like something that can be built up on, that could potentially lead to a therapeutic, but also just the process of going through, I don't know, the scientific method. I mean, it's, for me, it's like, ooh, I love it. It's my favorite.
I mean, I love it. It's like, I mean, it's very intellectually satisfying to me.
Yes.
But I will say that one of the reasons that it is that this is actually probably the thing that I'm like naturally talented at. It's talking and like communicating with people and writing. It's not science and virology. I love virology because I think it's beautiful, but I have to work hard at it. Like I can put my skills to use in virology, but like, and I understand pathogenesis really well, because let me tell you how I understand pathogenesis. It's a reality show. Alternatively, it's a football game. But it's a lot of different moving parts interacting with each other and just causing drama. Pathogenesis is fucking drama. It is abnormal inflammatory responses. It is the virus screwing with the host. It is the host like retaliating against the virus. It is constant like drama and like moves and counter moves and, all kinds of like plan B, C, D, E, F. And if you understand it, like nothing could be more interesting and like riveting. It's like watching literally an episode of Real Housewives or Secret Lives of My Wives. I'm serious. Like, I mean,
No, I
some
feel you.
viruses, you know why Ebola is like so bad? It's not because Ebola virus itself is so bad. It's because Ebola is like the equivalent of Taylor from Secret Lives of Mormon Wives, Taylor Frankie Paul, who is like probably the most naturally gifted reality star I've ever seen. She is just like the most televisually like engaging hot mess. And
yeah, that lady is a hot mess. I didn't finish the series, but oh my God, I'm like, wow, you are not really a Mormon.
okay, this is a sidebar that has nothing to do with virology, but I just have to tell you and everybody that my husband has been watching that show with me for 4 seasons. Sorry.
Oh,
I think
wow.
it's been
Okay.
fewer than 4 years, but 4 seasons. And only this season, with the terrible advent of the nuisance that is dad talk, did Alexi, my husband, discover that mom talk wasn't a bunch of moms talking. He's like, why are they always dancing? And I said, that's what mom talk does. Like they dance on TikTok. And he was like, they're not like exchanging parenting tips. It was so good. I was so delighted by that. But yeah, anyways, I mean, that's to me what pathogenesis is. And I think that one of the things I just find so rewarding is that the work itself is so important. The viruses that I work on are terrifying in some cases, like Ebola. Ebola works and is so terrifying, not because it's some sort of super virus,
It
but
isn't.
because it just is really good at subverting the host's ability to control its own responses to the infection. And it varies from host to host. Honestly, that's the research that got me any sort of academic faculty position at all showed. I got really lucky, and my work, which I think was important, was rejected by Science, which is a very highfalutin journal. And then they asked for it back when the West African Ebola epidemic happened. And so I got it published and in Science, and it was a hugely important career step for me. But it was also, at the time, this was really before everybody was into preprints and like there was Twitter, but not, 2014, not everybody even had a smartphone all the time. And we didn't have the capabilities that we have now. And so there was not really that much information to explain why Ebola was exploding in West Africa, far beyond what it had historically. Where did that virus come from? Was it, it had never appeared in West Africa before. This was over 1,000 miles from where the nearest Ebola outbreak had been before. Was it a new species? How did it get that far? Does Ebola have a much larger geographic range? Probably. Than we thought.
So.
And so getting my paper out at that time was an important contribution to the field. And so when you talk about stuff that is important, the model that I developed, even though people aren't always going to ask the same questions about pathogenesis as me, you can use the model that I developed to ask a lot of different questions. So it contributes to other people's research as well. And that's how the scientific community is operated. That's the importance of having journals and scientifically peer-reviewed literature, because we can build off of each other's work, but because truly AI cannot replace the experimental component at this point. There are far too many data gaps to even train the AI models, the most advanced models that we have, to even train them to correctly predict this kind of stuff. We are at a point where we have to be able to continue to do experiments. And the only way to do that is to publish our work. And then we build upon each other's work. And in that way, even if we're not directly collaborating, we work collaboratively as a community. And I think this continually gets lost, but this is one of the things I love the most about virology. Some of the most controversial, stressful work that I've been involved in, like the origins of the pandemic work that I've done, has been so rewarding personally and professionally, I've gotten a science and 2 Cell papers out of it.
Cell is also a highfalutin journal for the audience.
Yes.
So
and we've really developed, I've developed relationships with other scientists who are brilliant people and who are true friends, as well as collaborators and colleagues who have different experience and subspecialties and expertise than I do. And so in that way, I mean, we're really a community that does even more important work because we build on each other's work. And I think that this is something that continually gets lost as we have these discussions about vaccines. Oh, what do we do about this misinformation? Well, it's disinformation, first of all. The Health and Human Services Secretary, Robert F. Kennedy Jr., was on Capitol Hill all week last week. He's going to be back there next week. Me and Jessica Malati Rivera on our other show, Taking Shots, are going to be covering the Senate Health Committee meeting on Wednesday. So tune in for that. But, he's been all over the place lying. Like, there is not a lot of good sources. There are places like TWIV that you can go to. There are, media outlets that will only report accurate things, but there's not enough virology content, I think, that explains to people what our field's really about, what kind of a community it is, how important the questions we are, the questions we are asking are, the way that we can all build on each other and lift each other up for the good of everybody. The reason that we didn't have polio and measles and mumps and rubella and these horrible diseases that unfortunately we're all about to see again is because of virology research. And I think back, Juliet, I've been
it's so sad. Like, I mean, we came so far and here we are reverting. Like, it's my God.
We need to get back.
I mean, the number of people who are like rolling in their graves at this point. Like,
my man Jonas Salk, that's for sure.
Yeah, like Salk and Sabin and all these people, Kaprowski. Like, what? Like, how did this happen? I'm horrified, and I would never have expected this in this country, for real. Like, how are we moving away from public health and so drastically and so, I don't know, so sloppily? It's but
well, I think about this all the time, Juliet, and I think about it the way that we were trained to think about these kind of questions. I try to think about it as a mechanism, as a hypothesis testing to address a mechanistic question. How did this happen? How did all of this disinformation get to be so commonplace? Well, we didn't do a good enough job of pushing back against it. We can't control what the anti-vaxxers are going to do. And they've taken the opportunity to be very, very good at spreading a lot of disinformation.
Excellent stuff, man. They make a lot of great content. Like, I have had, like, during COVID, like, I had family members send me RFK Jr.'s documentary thingy. And I was like, guys, this is not real. Like, this stuff, none of this is correct. And they're like, well, you're a part of that system too. So like, they've done a very good job of riling people up for like these false sort of narratives. But
it's a propaganda campaign.
Yes,
it's
exactly.
Far beyond at this point riling people up and just sowing political division. I mean, at this point, it's coming from the government, and it's incredibly alarming. It's essentially a cornerstone of fascism and authoritarian governments. And the idea that this man, who is unbelievably unqualified to be in charge of anything, much less people's health, is calling the shots is insane. And part of the reason that we are in this situation is that we can't control what he's going to do. They've built huge, media networks. They've got content, as you mentioned, during the pandemic. They've been building that entire network and system for disseminating propaganda for decades. And they were waiting for nothing but the pandemic. When it became something that was in the Trump administration's political interest and the Maha movement's political interests, then, it really, really gained the ability to, I guess, achieve escape velocity after they had laid this incredible foundation. So the conclusion that I've come to is that we can't control any of that. But what we can control is how we respond to it. And we have not done a good job of that, because there are a lot of people that want to hear about this or that paper and this or that new achievement in science. And I think it's important that we continue to have content that is just strictly scientific and that goes that way. But I also think we really need to do a better job of explaining this stuff to people.
Oh, absolutely.
Why the hell was Jonas Salk like Times Man of the Year? He was on the cover of every single newspaper for weeks. I mean, he was an American hero.
Absolutely. I think putting
it was not a question in anybody's mind. How do we get back there? We need to engage people, and we need to tell people like that we are here to help them. We don't need to sit here and take these attacks from these anti-vaxxers lying about us and our field and the science that we do. We need to stand up for it, but we need to also get people on board so that they're not bored out of their minds listening to us talk about it.
Communicating, yes. Like we need to do that. I agree. So how are we going to do that? Is this part of the plan? Like we are going to do our best to make sure that science is engaging as we talk about it every week or every month. I don't actually even know what our format is.
I don't know what our format is either. I mean, I think we're kind of experimenting with it and finding out what works. But my suggestion, Juliet, would be also, because it's not that we're lazy. We're actually both incredibly hardworking.
Very busy.
But we aspire to be lazy, and we're both overworked. And so we don't want something that's going to become a burden. But we do, I think, I don't think that us getting up and giving a PowerPoint presentation or doing a journal club every week or other week is going to be something that's going to be that gripping to a lot of viewers. And you know what? I'm not going to remotely ever try to compete with Vincent as like the virology podcast to go to if you want, a casual but very serious, like multidisciplinary panel discussion, like sort of like the McLaughlin group for those of you who are really old. About viruses and virology. Like, I think Twiv and Vincent have that covered. Like, I don't need to be teaching people about virology. Like I said, that is hugely important. But I think I can reach a different market. And I think you can too. Because one thing that we've always been great at, Juliet, is getting together to talk shit and be funny about it. And everybody has always, as we were saying at the beginning, we've had a reputation, whether you believe it or not, as being super fun. And part of it is not just because, we get free pizza and beer, at least I did through political means. If you count the graduate student organization of the Columbia University Medical Center integrated program to be political means. But technically it was. But anyways, I think that we've always had a reputation as being two people who were fun and interesting to hang around with. In fact, I was complaining that I aspire to be really boring and to do nothing all the time. And I no longer have the luxury of doing that in one of the chat groups that I'm in. And somebody that we went to graduate school with is in that chat. Mm-hmm. And they said to me, Angie, there's many things about you. He's like, but you're never boring.
That's for sure.
And I think that together, that is very true. Like we were just like a freaking hurricane of fun times, including our discussions about virology. Mm-hmm. And because of who our mentor was, we were trained to be very rigorous virologists in the old school fashion. I mean, my trainees have been shocked that I didn't kill everybody because I have cesium chloride gradient and the plasmid
and that we know things. Like I'm so surprised. Like my students are like, "Oh, you can teach me." Yes, I can teach you stuff. That's my job.
Yeah,
I can be in lab. They're like, talk to Morrison, Juliet. Like, what are you doing in lab? An experiment?
Well, I have found that things go wrong in lab when I'm in there. It's very annoying for everybody else. So I avoid it.
No, man, I actually love lab. I wish I didn't have all these other things like kind of weighing on me, which is another thing that the public does not understand, that like people who are in like academic science, we are doing so many different things. We're teaching, we're running a lab, which is basically a small business, and we're also like have to do so much service to make sure that we're putting science out there publicly. I don't think they realize just how committed an academic scientist is. Like we are here because we care. We're curious, but also we want to get the knowledge out. Some of us are not the best communicators, but I don't think that means that we should like cut people off. The other people who are very slick on the other side they're the ones who are like manipulating people. Like we actually have the knowledge and the training to give real advice and guide people forward. Like I don't know, I'm not going to take my biomedical advice from a lawyer who had like something in his brain. Like I just can't. Like
Oh,
I would
you
rather.
mean Aaron Siri, like the ambulance chasing lawyer who's RFK Jr.'s surrogate, who constantly wants us to get rid of the vaccine injury compensation program so that he can make literally hundreds of millions of dollars by putting vaccine companies out of business and thus depriving all of our children of their vaccines. Yeah, I'm not going to trust them either. But here's the problem. I mean, we haven't done a good job of communicating.
No.
And I think, a pretty good communicator, and you are an excellent communicator as well. Why don't we just communicate differently? Why does science #SciComm have to be like, either so simplistic and dumbed down, or it has to be like, oh, we're going to empathize with everybody. We're going to be nice to the Maha people and work with them collaboratively. We don't want to condescend to them. No, I'm going to call liars liars. I'm going to call things like they are, exactly as I've always been doing. And Juliet, you and I have been having these kind of conversations, candid conversations that are based in evidence and science for years. We're also hilarious. We are funny. And yeah. I think that, we should just have more conversations about this stuff, because frankly, at the very least, even if nobody watches, it feels very cathartic. And I feel like I can use all of the mentally therapeutic activities.
Yes.
I know both of us have struggled sometimes with chronic illness too. And I'm currently dealing with some medical issues that have been fairly serious and are still pretty unresolved. And with all of this, it's hard to think like, oh, I'm going to now have a whole separate science communication thing where I'm going to be making presentations and fancy things. I mean, I'm like very bad at making influencer videos, Instagram reels, which I'm trying to do despite the fact that I'm like an old lady. So I don't have time to become like a professional influencer, and that's not what I want to do. But I do want to provide people with other opportunities to get a window into what it's like to be a virologist right now.
I agree.
Because it's all I think about. It's consuming me. It's consuming everybody I know, and it's consuming our profession. And part of me wants to just cry for help from the public. But part of me also knows that we just didn't do a good enough job of standing up for ourselves. And we need to do that now. So rather than like whine about it, which I just only do to my husband and my friends like you, I'm going to try not to do that in these conversations. I'm going to try to talk about what I love about virology and why. Why am I continuing to put up with like literally massive campaigns from Canadian ostrich fascists to have me fired? And that's a topic for another show. But like we both are putting up, and everybody in our field is putting up with so much pressure, whether it's public pressure. And I mean, mine is of course extremely dramatic because I'm a drama queen, but everybody, even people who are dealing with mundane drama, that's even worse. If it's, I'm going to lose all my grant money and I have to my lab down. And we are just in a position that's so precarious. I don't know what so precarious.
I don't think people understand that unless they are directly related to a virologist or a biomedical scientist or scientists in general. They don't realize just how much of the operation works, and the fact that we are now being crippled. And there's like I don't know. Like I tried, like I saw the writing on a wall, and I was like, oh, let me start. Like I'm gonna do a GoFundMe or some sort of thing, and I put up together like this thing for UCR's Give Day. I got $660. I mean, that's something, right?
It's something.
Like, I'm really happy to the people who did give, but like, like that could probably buy an antibody and a half for me. So like it makes
buy you two antibodies from Santa Cruz, it won't work.
Thousands, and we're not getting that anymore. Like we are under serious attack. Nobody cares about viruses. They have like deprioritized infectious disease, and like that scares me because we are living in a situation right now where no one's tracking things. A lot of people don't trust the public health or or scientists, and so I see it. I see an explosion. Like it's already measles. Like we're gonna have like German measles now. Rubella. Like what's next? It's crazy.
Well, there's already increasing mumps. There's been increasing cases of infant tetanus, which, I mean, I
didn't even know that was a thing.
It is a thing, unfortunately, and because Clostridium tetani is in dirt, so babies are also more susceptible to it. Tetanus is a toxin that's caused by a soil bacterium, so it's only a problem if you get enough of it embedded, which is why it's a problem with puncture wounds and things like that. It's not really. It's infectious in that it's caused by bacteria, but it's not infectious. You can't transmit it back and forth. It's an opportunistic toxin, really. And there's just no reason for infant tetanus to be a thing. Tetanus to be a thing. I mean, that's what the DTaP is for. Pertussis there have been multiple pertussis outbreaks in multiple states. I mean, we are in a really difficult position for society, and but
you know what's crazy? Juliet, like Angie, you know what's freaking crazy? That the same people who would be like, oh, we're not going to vaccinate our kids because we don't understand the science and this all of that. They're the same ones who are going to go and get Botox and filler. So like, you don't mind putting things that you don't understand into your body. You just somehow want to draw the line at vaccines. You're putting a freaking toxin. Botox is a freaking toxin. And don't get me wrong, like I have actually used Botox myself. Like I use it for my like migraines and stuff. But
I totally use it for my face as well as Juvederm. I'm a big fan.
But it's like how do you pick and choose this? We think vaccines cause all these problems, but Botox is okay.
Because Juvederm
is okay. It's so silly.
Most of the people who are choosing it are not choosing it, and that's what the problem is. They're being coerced into believing it because they are the targets of a propaganda campaign. And so that's why I think, and we're going to wrap up. I think, we should do this once every 1 to 2 weeks to start and see if people are sick of us at that frequency. And if they demand more, we'll give them more. And if they demand less, then we'll give them less. Yeah. But I think that for me anyways, like one of the things that I've really, really struggled with is just not being overwhelmed by despair and trying to kind of keep hope alive and thinking of something positive to do. So granted, I find it very positive to focus on how I'm going to destroy all of my enemies who are doing this to us and who are traitors to the US and our American values, as well as my values as a scientist. And so that's something that motivates me. But besides that, what else can I do besides, plotting revenge eventually and thinking about how we might rebuild once these people are out of power? And I think the most important thing we can do now is to lay a foundation of truth, give people a reason to trust us. Give people a reason to remember that we're human beings and that there was a reason why this was bipartisan, why the U.S. led the world in funding research, why the U.S. led the world in innovation, why the
Absolutely.
US economy was one of the strongest for generations.
And why people have been coming to America forever. Like I myself, I'm an immigrant. Like I came to this country because I was like, this is where you're going to do the best science. And that's what I felt within my heart. And for the longest while, that is exactly what it was. And I will tell you, recently, like I'm on the admissions committee for the grad program for my department, and we had some candidates. Sometimes you have candidates reach out to you before they apply, and there was this one guy who was so promising. And at the end of it, he was like, "Nah, I think I'm just gonna apply to Europe." Like he was like a Chinese student, but he was living in the States even. But he's like, I just don't want to do this anymore. So like we've managed to like scare off like somebody I thought was quite talented. I was like, my God, if I could get this person in my lab, I'd be so happy, because he seems to really understand the repercussions of things and really understands like how to do science. Yeah, we're losing all of that. So like I was attracted to this country because specifically because I wanted to do research and science, and I don't know, like I had these big dreams, and I know a lot of people still feel that way, but like we are having a hard time doing that. Like I would say to the public, like right now it's really rough, because like every day the rules change, and there is no money for some of the most pressing issues at this point.
And I think what's happened, Juliet, is that, I mean, throughout the federal government right now, what is happening is that a lot of the mechanisms being used to destroy science, but throughout the government are really boring. I mean, people, when something's happening in a policy advisory meeting where the results aren't immediately apparent, it's easy to understand why war in Iran is bad and dangerous. It's a lot harder to understand how a recommendation that's made by an advisory committee for immunization practices, or ACIP, that's full of anti-vaxxers is dangerous. But the ACIP recommendation will kill far more people and create much greater dangers to our economy and our national security. And it's just very difficult to explain this to people, that when you're cutting grants, you're not just putting like a scientist out of a job and, oh, now you got to go get a real job. Like
Right.
you can't screw around your lab anymore. No, they're actually not investing in their own futures and the future of our economy, the future of all of our healthcare,
Absolutely.
their own children's future. That's what people are saying no to. And in my view, it's not something that most reasonable people would say no to. And so the only conclusion I can come to, based on the evidence that I've seen, and we've all seen it, is that people have been lied to.
Exactly.
And that's why they don't
just
trust
don't
us.
know. Yeah.
And people have been saying, oh, you were wrong about COVID, like in January of 2020, or you didn't like masks at first. I didn't think that masks were going to work. And they don't fully work. Like everything is on a gray scale.
I have always said, guys, don't listen to anything that these people are saying. Wear your masks, because like physics.
Well, I mean, but Juliet, like at the very beginning of that, like March of 2020, I was like, I don't know how good masks are going to be. And people now are still saying like that nobody can trust all of public health because like I was wrong and I changed my mind about that. Or Fauci was wrong once, or told, he did say that about masks and it was for the wrong reason and stuff. That is not why the public isn't vaccinated.
We make mistakes, and that is okay if you then self-correct, admit your mistake, and move on. And the fact that like the average scientist will be like, oh yeah, my bad, I am going to walk that back and I'm going to fix this, should give people a lot of I don't know, like you should feel good about this. Like the scientists we don't want to mislead people. Like I certainly don't want to mislead people. So the minute I realize that something's wrong, I'll be like, man, sorry. Like I interpreted it wrong.
And that's the thing, Juliet. And we're gonna I think we should close up, because as always, we've gone over time. But what I was just gonna say, my last statement on all of this stuff, is that the public did not lose trust in us because we were wrong some of the time. and we made mistakes and we're human beings. The public doesn't trust us because these people have been lying about us and they have dehumanized us to the public. And so that's why I'm really glad that we are going to have these conversations regularly. Please give us your feedback and let us know what you think and what you like and what works for you and what doesn't. Because one thing I think that, like I said, Juliet and I can do very well together is just be fun and funny and have like a fun conversation about a topic that we both have expert level knowledge in, even if we're annoyed about it, like I am about these dumb Paramyxoviruses that I shouldn't have to know about. But I just really want to thank you guys for sticking in there. And, on the health site, we have almost 700 people watching us. So, it's pretty exciting.
That is super exciting, dear Lord.
I'm just really grateful for everybody who shows up to listen to me and my friends talk on my Substack or whatever platform you are watching on. Please do go check out my Substack, rasmussenretorts.substack.com. I'm helping Professor Morrison here develop a more expansive social media profile. So stay tuned for that.
One day.
But we'll see you back here in a week or two. And I'll let you know. I've got all kinds of fun marketing material ideas to share with you. So thanks everybody so much for sticking in with us, even though we went a little bit over time.
Yes, thank you so much.
Great seeing you, Juliet, Julie Poo.
Great to see you too, Angie. Angie.
It's so good to see you. It's so good to be talking viruses with you. So can't wait till next time. All right, take care.
Bye-bye.